Chapter 49 is on the nervous system, and we're going to go through 49.1, which talks about neurons and nerve impulses. So we looked at this yesterday. These are some. This is a picture of some nerve cells. And we filled this out yesterday. We talked about the cell body, which contains the nucleus, which is right here. And we talked about the dendrites, which is like the inbox. It brings information into the nerve body, which then goes out. You can say the outbox, which is the axon. And the axon will send that message down to the terminal end, which will be attached to either, I won't say attached, but it will then either conduct an action potential to the next neuron, or it will turn on or a muscle, so it can cause a muscle contraction, or it can send a signal to an organ. We talked about this lipid coating called a myelin sheath that insulates the axon, and that is produced by the Schwann cells. We really didn't have much time to talk about the action potential, but the action potential is the actual signal that's coming into the nerve body and then out. And we talked about how that signal, if it jumps the gaps, which is the nodes of Ranvier, it can go faster. So this is called the synapse. The synapse is where the actual signal is being transferred either to the next nerve or a muscle or an organ. So when we look at this, we have what's called the presynaptic neuron and the postsynaptic neuron. So here's the pre, and this is the post. So what we have here is a signal that's going to come down to this axon, and then we've got vesicles here which contain neurotransmitters. That signal, that action potential that comes down, is going to tell those vesicles to fuse with the membrane here on the axon and to deliver its contents through exocytosis. So it's going to exocytose these neurotransmitters into this, what we call the synaptic cleft, this space. Those neurotransmitters will then go through these little channels and cause a new action potential to occur and send that signal to the next nerve, muscle, or organ. This is important because, like I was talking about in class, if the nerves are physically connected, as soon as we turned one nerve on, all of the nerves would then be turned on. So we have to have a way of controlling the signal from one nerve to the next, or from the nerve to the muscle and the nerve to the organ. And this is how it's done. So let's talk about a membrane potential. And when we talk about potentials, what we're looking at here is the potential to do work. So what we have is we have a cell membrane. So the cell membrane is right here, and you see our phospholipid bilayer. And we have a channel here. Now, these are ion-gated channels. They're very specific to what they will let through. They also can be open or closed. Now, if you look at this partition in which we have, this is outside of the cell, and this is inside of the cell. Now, when you look at this, you'll see that we have a lot more negative. We have this negative proteins here that create a negative charge on the inside of the membrane. And then we have a lot of positive, our sodium, on the outside, which gives us a positive charge. Notice the ion concentration gradients. So sodium here is high on the outside and low on the inside. So it's going to want to move in this direction based on diffusion. Remember, diffusion does not require energy, but we have these concentration gradients, so it's going to want to try to balance itself out. So sodium wants to get into the cell. On the other hand, we have all this potassium on the inside of the cell versus the outside. So again, we have a different concentration gradient based on that particular ion. And it's going to want to move out of the cell by diffusion. So we have a situation here where we have a lot of potential to do work in order to, and that work would be to move across the membrane. So we want to move potassium out and sodium wants to move in. Because of this situation, the inside of our membrane has a negative charge and the outside has a positive charge. So in order to maintain what we call our resting potential, we have some of these channels that will leak. So right here, this one is just for sodium. It will leak in sodium. Over here, it's going to leak out potassium. And again, this one's going to leak in potassium. And this is all going to be based on concentration gradients. All right, so we have these ion channels that, if open, can move these ions across the membrane. But right now, we want to get to what's called our resting potential. Now, this one right here, this is a pump. Now, this pump is going to establish those concentration gradients by active transport. And remember, active transport requires ATP. So this little guy right here is going to be trying to maintain that resting potential where these other pumps will be used for sending what's called an action potential. All right, here comes the craziness now. Let's go through this. This is going to be the hard thing to understand. So let's start with number 1 over here in the bottom left-hand corner. This is the resting state. So remember I talked about those ion channels. Notice they are closed. The inside of the cell has that negative charge and the outside will have the positive charge. Positive charge. And that was all maintained by that sodium-potassium pump. So we have our resting potential, which if we look is right here on our middle diagram. The next thing that we're going to have happen, to get this back on the center, is we're going to have a depolarization. So we'll have some kind of stimulus that opens up the sodium channels. So notice right here, our sodium can get in. All right, the potassium still is kind of stuck. All right. Can't go anywhere. But the sodium can come in. And as the sodium comes in, remember sodium has a positive charge, it's going to depolarize. It's going to cause depolarization in that it's going to change because we were down here. If we look at our original chart, notice how we're in the negative when we start off. As the sodium comes in right here, as it comes in, okay, we're starting to get more positive, right? As more sodium comes in, then we're going to move to the next stage, which is up here, stage 3. And that is going to cause more sodium channels to open and more sodium is going to flood in, which is then going to make our potential go even higher in the positive till we finally get to what we're looking for, and that is our action potential. Our action potential says, okay, we're on. And once we're on, that's where the signal is sent. Ugh, this pen is going to drive me nuts. This is when the signal is on. So this is our on. So it sends that signal. It causes those vesicles to fuse with the presynaptic membrane, dumps the neurotransmitter, and sends the signal to the next nerve. Once that signal is on, the next thing that's going to happen is we're going to have the sodium channels, notice they close. So once the sodium channels close, we're going to have that potassium is still open, so potassium is going out, and this is causing what we refer to as our repolarization, so repolarizing our membrane. Now, sometimes this takes a little too long and too much potassium goes out, and so we dip down a little bit too far. And then again, the sodium-potassium pump will fix that, and then we're back to our resting state. So this is what we're trying to get. We're trying to get to this right here, that on switch. So once we're on, then everything closes back up, and we reestablish our resting potential. If we do not reestablish our resting potential, we can't send the next message. So this is happening very quickly, so you can send one message after another. So think about when you're contracting your muscles, how fast this process has to take place. Now, this is a video that will go into this in a different manner. I would highly suggest going onto Canvas and clicking on the teacher lecture, and then you can kind of flip through the slides to this one and then click on that and watch the video. All right, so when we look at this picture here, we can see up here the synthesis of a neurotransmitter, which is taking place up in the cell body. Those vesicles then move down the axon, and then as they're sitting down here waiting, once we have our action potential moving down, the action potential will get down here on the presynaptic terminal. And actually what I'm not showing here is there's a calcium channel there. That action potential will open the calcium channel. The calcium will come in, and that signals those vesicles to fuse with the presynaptic membrane and to exocytose those neurotransmitters into the synaptic cleft. The neurotransmitter will then combine with these receptors on the postsynaptic cleft, and then it will relay that on signal to that nerve there. Now, we don't want the neurotransmitter to permanently bind to the receptor, because then that receptor is no longer going to be able to send an action potential. So they'll bind and let go and bind and let go. But once that signal's gone, we're going to reuptake. We're going to take that in or break it down, one or the other. We'll either recycle or break it down so that we don't continually have this particular neuron stimulated. Right, so when we talk about these neurotransmitters, again, they are what is kind of like relaying that message from one nerve to the next. Here is another video that I highly suggest you watching, and it talks about how your body reacts to external stimuli. So go again back to Canvas and watch this, I think it's like 4-minute video. And here I have a blank nerve so that you can practice labeling the parts, because you will need to be able to do that for your test.